developing in vitro release testing methods for parenteral suspensions. Our presenter, Dr. William Smith, is currently an ORISE Fellow with Dr. Xiaoming Zhu and the Division of Product Quality Research, working on complex drug formulations from emulsions to implantable polymeric devices. His research focuses on the physicochemical characterization of nano and microscale materials, including the development of analytical and in vitro release test methodologies to support assessment and review, equivalence determination, and evaluation of drug product quality. Our final presentation in this session is on MAPP 5019.1, allowable excess volume and content, and injectable drug and biological products. Our presenter, Dr. Hannah Wang, is an experienced policy lead and CMC reviewer in CEDAR OPQ. She's led multidisciplinary cross-center working groups in developing internal and external policies, including the training programs for the implementation of new policies. Please join me in welcoming our first presenter, Dr. Shenrun Li.
Good afternoon, everyone. My name is Shenrun Li, a pharmacologist in the Division of Bioequivalence II, within the Office of Generic Drugs. It is my great pleasure to share with you my knowledge on the Q1-Q2 assessment and the regulatory pathway for Biowaiver of injectable solutions. Before I go into my presentation, here is my disclaimer. This presentation reflects the views of the author and should not be construed to represent FDA's views or policies. In this presentation, we'll learn the following objectives. First, I will briefly introduce the Code of Federal Regulations related to the biowaivers for injectable solutions. Then, we'll learn assessment using appropriate regulations through case studies, including considerations for differences in exception excipients, considerations for differences in non-exception excipients, consideration for different dosage forms and different strengths, and considerations for multiple administration routes. I will start off by introducing the Code of Federal Regulations related to the biowaivers for injectable solutions. Regulations at 21 CFR 320 address the requirements for bioavailability and bioequivalence data for approval of NDAs, ANDAs, and supplemental applications. Provision for waivers of in vivo bioavailability and bioequivalence studies under certain conditions is provided at 21 CFR 320.22. Under 320.22b, for certain drug products, the in vivo bioavailability or bioequivalence of the drug product may be self-evident. FDA shall waive the requirements for the submission of evidence obtained in vivo, measuring the bioavailability or demonstrating the bioequivalence of these drug products. A drug product's in vivo bioavailability or bioequivalence may be considered self-evident based on other data in the application if the product meets one of the following criteria. The drug product is a parenteral solution intended solely for administration by injection, or an ophthalmic or otic solution, and contains the same active and inactive ingredients in the same concentration as a drug product that is the subject of an approved full new drug application or abbreviated new drug application. Code of Federal Regulations, Title 21, Section 314-94, provide requirements or basis for an ANDA submission. The regulations at 21 CFR 314-94-A93 identify permissible differences in certain inactive ingredients for drug products intended for parenteral use, commonly referred to as exception excipients, if the ANDA contains sufficient information to demonstrate that any differences do not affect the safety or efficacy of the drug product. For example, drug products intended for parenteral use generally must contain the same inactive ingredients in the same concentration as the RLD. However, an applicant may seek approval of a drug product intended for parenteral use that differs from the RLD in preservative, buffer, or antioxidant, provided that the applicant identifies and characterizes the differences and provides information demonstrating that the differences do not affect the safety or efficacy of the proposed drug product. Intended to give applicants flexibility to seek alternative ways of complying with the statutory standards for drug approval, the agency has codified a waiver provision applicable to ANDAs at 21 CFR 314-99B, under which an applicant may ask FDA to waive under this section any requirement that applies to the applicant under 314.92 through 314.99. The applicant must comply with the requirements for a waiver under 314.90. If FDA grants the applicant's waiver request with respect to a requirement under 314.92 through 314.99, the waived requirement will not constitute a basis for refusal to approve an ANDA under 314.127. Now, let's learn more on the assessment considerations through case studies. I would like to note that the hypothetical examples in the next few slides are provided to highlight some of the agency's considerations for evaluating a waiver request during the scientific assessment of an ANDA. Examples are intended to illustrate instances and do not constitute grant scenarios. A decision to grant a waiver request is based on scientific evaluations of the specific facts of an individual ANDA, including the supporting documentation and the justification submitted for the specific waiver request. Case 1 for Q1, Q2 assessment for exception excipients. In this case, the proposed test formulation is not Q1, Q2 to the RLD in exception excipients. For the comparison table, the RLD contains sodium citric as buffer and the sodium sulfate as antioxidant, while the proposed test formulation contains mannitol as buffer and does not contain antioxidant. For Q1, Q2 assessment consideration, for the regulations at 21 CFR 314-94-A93, a variation between test and RLD products is allowed for parenteral formulations with respect to buffer, preservative, and antioxidant, provided that the applicant identifies and characterizes the differences and provides information demonstrating that the differences do not affect the safety or efficacy of the proposed drug product. Additionally, the buffer, preservative, and antioxidant amount in the proposed test formulation should be below the levels in FDA-approved drug products for the same route of administration and context of use. Combining the consideration above, the differences on exception excipient in Case 1 are acceptable under 21 CFR 314-94A93. Case 2 for Q1-Q2 assessment with ROD formulation change. In this case, the ROD formulation was changed in non-exception excipients, and the proposed test formulation is Q1-Q2 to the original ROD formulation. From the comparison table, the current ROD formulation contains tartaric acid and dihydric disodium acetate, but neither of the inactive ingredients presents in the original ROD formulation or the proposed test formulation. Both inactive ingredients are not exception excipients. For Q1-Q2 assessment consideration for this case, a 314-99B waiver is appropriate only when the ROD formulation change is not for safety or effectiveness reasons. Biowaiver is granted under 21 CFR 314-99B for Case 2. Case 3 for Q1-Q2 assessment when ROD and RS are different. In this case, the proposed test formulation is not Q1-Q2 to the ROD in non-exception excipients, but Q1-Q2 to the RS. For the comparison table, the ROD contains sodium chloride as isotonic agent, but the RS and the proposed test formulation contain sodium citric as the isotonic agent. For Q1-Q2 assessment consideration, a 314-99B waiver may be considered with supporting documentation for the request. Please note that the acceptability of a request for waiver under 21 CFR 314-99B will be determined based on scientific evaluation of the specific facts of an individual ANDA. Case 4 for Q1-Q2 assessment for dosage form change. In this case, the proposed test formulation is Q1-Q2 to the ROD formulation with different dosage forms. For the comparison table, the ROD is a solution for injection, while the proposed test drug product is a powder for injection. For this particular case, an approved suitability petition is required for the change of dosage form prior to the ANDA submission per the regulations at 21 CFR 314-93. Case 5 for Q1-Q2 assessment for strength change. Similar to the previous case, the proposed test formulation is Q1-Q2 to the ROD formulation with different strength. For Q1-Q2 assessment consideration, an approved suitability petition is required for the change in strength prior to the ANDA submission per the regulations at 21 CFR 314-93. A 314-99B waiver may be considered for this particular case. Case 6 for Q1-Q2 assessment for multiple administration routes. In this case, the proposed test formulation is Q1-Q2 to the ROD formulation. Both ROD and the proposed test product are for multiple administration routes, including intravenous, intramuscular, subcutaneous, and endotracheal. For this case, drug products with multiple administration routes can be evaluated for bioequivalence under different regulatory provisions. In vivo bioequivalence study requirements for the intravenous, intramuscular, and subcutaneous routes of administration may be waived under 21 CFR 320-22B1. And for the endotracheal route of administration, FDA can evaluate bioequivalence using in vitro approaches under 21 CFR 320-24B6. Case 7 for Q1-Q2 assessment for special administration route. In this case, the proposed test formulation is Q1-Q2 to the ROD formulation, and both drug products are cardiac perfusions. For Q1-Q2 assessment consideration, regulations at 21 CFR 320-22B1 can be utilized as cardiac perfusion can be considered parenteral solution for infusion. Now we have went through all the case studies, and it's time for the challenge questions. Challenge question 1. Differences between the proposed test and ROD formulations for exception excipients in a parenteral drug product may be acceptable under A. 21 CFR 314-99B, B. 21 CFR 320-24B6, and C. 21 CFR 314-94A93. The answer is C. Under 21 CFR 314-94A93. Challenge question 2. Which of the following statements is not true? A. As long as the proposed test formulation is Q1-Q2 the same to the original ROD formulation, waiver request can be granted per 314-99B. B. 314-99B waiver can be granted when test formulation is different from the ROD formulation in non-exception excipients. And C. Drug products with multiple administration routes can be evaluated for BE under different regulatory provisions. The answer will be A. Even if FDA grants a waiver of a requirement in 314-92 through 314-99 in a particular application under 314-99B, such as the parenteral test formulation is the same as the previously marketed ROD, the application still must meet all applicable requirements regarding safety of the inactive ingredients for approval. And this concludes my presentation. With that, I would like to thank all my colleagues in the office and the Division of Bioequivalence, especially Dr. Zhang, Dr. Park, Dr. Miller, Dr. Li, and Dr. Murphy, and my colleagues in the Office of Generic Drug Policy and the Office of Research and Standard. I would also like to thank all who has contributed directly or indirectly to this presentation. Thank you all for listening. If you have any question, I'm glad to answer in the panel session.
Thank you for the introduction. Hi everyone. My presentation today is about current thinking and research on in vitro only approaches for injectable drug substance suspensions. There are three learning objectives for this presentation. First, to identify injectable drug substance suspensions. Second, to categorize the injectable drug substance suspensions into three groups. And third, to provide a summary about the current thinking and research regarding in vitro bioequivalence approaches for each of the drug substance suspension groups. Here I would like to first provide some background information about injectable suspension. Injectable suspension are suspension products for parenteral route of administration. The most common injectable suspensions are drug substance suspensions. It's a relatively simple system. The particles within the drug product are made up only of micro-sized or nano-sized drug substance, and there are no release modifier, no insoluble excipients in the solution phase. The Orange Book also lists some PLJ polymer-based microparticles and in situ forming implants as suspension. These products are not considered as drug substance suspensions. They are out of the scope of this presentation. Compared to suspension products containing release controlling excipients, drug substance suspensions have simpler release mechanism in terms of the following aspects. Drug substance particles are the only insoluble components in the formulations. As there are no other release controlling excipients, in general, the dissolution rate is generally determined by particle size and solubility of the drug substance. Based on drug release feature, duration of use, and dosing interval, and indications, we can roughly categorize injectable drug substance suspensions into three groups. Group 1, immediate release suspensions. Group 2, injectable suspensions for short-term use. And Group 3, injectable suspensions for long-term use. Both Group 2 and Group 3 products have certain level of extended release feature compared to Group 1. I will provide some examples for each groups in the later slides. Before diving into the examples for each of the group, I would like to talk a little bit about bioequivalence approaches for injectable drug substance suspensions. Bioequivalence studies should be sensitive, accurate, and reproducible. For systemically acting injectable drug substance suspensions, FDA generally recommend in vivo B study with pharmacokinetic endpoints in a product-specific guidance. In addition to in vivo B studies, FDA has recommended in vitro B studies for some of the injectable drug substance suspension products. An in vitro only B approach generally relies on totality of evidence. The principles is when a suspension product is formulated Q1 and Q2 the same, there could be difference in the arrangements of matter within the dosage form, which may impact product performance. The differences in the arrangement of matter may arise from difference in manufacturing, processing, or excipient grade and source. The differences can be evaluated by comparative physicochemical tests and in vitro performance test. The similarity in physical chemical characteristics and in vitro performance can demonstrate overall product sameness and equivalence. This slide provides some examples for Group 1 immediate release injectable suspensions. The first example is azacitidine powder. It's indicated for the treatment of patients with specific FAB myelodysplastic syndrome subtypes. It can be used for either subcutaneous injection or intravenous infusion. When it's used for subcutaneous injection, the product is reconstituted in 4 ml water for injection to get a cloudy suspension for subcutaneous injection. When it's used for intravenous infusions, the product is reconstituted in 10 ml water for injection. In this case, the whole vial of product is fully dissolved as a solution. The second example is meloxicam solutions. This product is indicated for the management of moderate to severe pains. The dose interval is once daily, and it's also for intravenous injection. The third example is dantrolene sodium for suspension. It's indicated for the treatment or prevention of malignant hypothermia. It's for single use only, and it's also for intravenous injection. Both meloxicam solution and dantrolene sodium for suspension are nanocrystal suspensions designed to have rapid dissolution after intravenous injection. Here we can see there are some common features for group 1s. All of those products provide immediate systemic availability of suspended drug. In the case of dantrolene sodium and meloxicam injection, they both have rapid dissolution after intravenous injection. In the case of azacitidine injection, because it has relatively high solubility in water, it's fully dissolved prior to intravenous injection. And all of those products has a relatively short dosing interval, or it's only used for one-time use, and they are all for short-term use. It may be feasible to use in vitro studies to demonstrate bioequivalence. In fact, FDA has recommended in vitro the options in the product specific guidance for those products. Here are some additional considerations when using in vitro studies to demonstrate bioequivalence of immediate release injectable suspensions. In general, when IVRT is used for bioequivalence purpose, the applicant should demonstrate the method is discriminative to the effect or process variability in production of the test formulations. Here, because those products is intended for rapid dissolution after injection, the discriminated power of IVRT may not be practical for this type of products. The main purpose of the IVRT is to show the test products and the reference product have comparable fast release. The applicant should still provide proper justifications in the IVRT method development and validation reports. This slide provides some examples for Group 2 injectable suspension for short-term use. As you can see here, the examples include several steroid injectables products and antibiotic injectable. They have generally lower solubility than the products listed in Group 1, and the particle size distribution of these products are in general in micron size range. Those products, most of them does not have a well-defined dosing interval in the drug labeling. But the suspension can provide a certain level of extended release feature after injection. Here are some common features for Group 2 products. They are micro-size, solid particle suspensions with certain level of sustained drug release, and they do not have well-defined dosing interval, and they are not for long-term use for chronic disease. As we can see here, they are mostly steroids or antibiotics. It may be feasible to use in vitro studies to demonstrate the bioequivalence of these products. In vitro B option or in vivo B option are recommended in the product specific guidance for those products. For example, triamcinolone acetone injectable injection, methylprednisolone acetate injectable injection, and penicillin G benzothine injectable injection. Here I would like to provide an example of in vitro B options. The example provided here is from the product specific guidance of triamcinolone acetone injectable injections. This in vitro B option is based on formulation Q1, Q2 the same to the RLD, acceptable comparative physicochemical categorization of the test and the reference standard products, and acceptable comparative IVRT from the test and reference standard products. Medical research has been conducted to improve the understanding on how flocculation properties of injectable suspension impact product categorization, dissolution profiles, and potential PK properties. Triamcinolone acetone injection was used as a model drug in an FDA internal project. It was found the suspension is a flocculated system containing primary particles of about 2 microns and secondary particles of tens of microns. The conversion between flocculate and deflocculate were highly shear dependent. Stirring speed and sonication had an impact on particle size measurement. In addition, sample introduction method that involving different shear also had an impact on the particle size measurement. A discriminative IVRT method was developed in this project. The deflocculated particles had 5 times higher dissolution rate than the flocculated particles in the initial phase. This result also indicated that if similar shear-induced changes during injection happened in a clinical setting, it could have implication for drug PK profile. Next, let's take a look at examples for Group 3, injectable suspensions for long-term use. In this table, we have a couple of antipsychotic injectable suspensions, and one for HIV treatment, one for HIV prevention, and one product for pregnancy prevention. All of these products have well-defined dose interval from 2 weeks to 6 months. They are all for intramuscular injection. Here are some common features for Group 3. Those products, they are nano to micro size solid drug particles for intramuscular injections, and they are intended for extended release of drug. This is also clearly indicated in the drug labeling. They have well-defined dose interval from weeks to months, and in general, they have indications for long-term therapy or for chronic disease treatment. Currently, the posted product-specific guidance for products listed in Group 3 recommend in vivo B studies. From formulation standpoint, Group 2 and Group 3 products are similar. However, Group 3 products do have a higher risk of clinical failure, given that they are for long-term use. Although In vitro B approach may be scientifically feasible. Additional information may be needed to support an in vitro B approach for group 3. For example, you may consider using PK or IVIVC to support design space of the selected critical quality attributes proposed in the in vitro B studies. GDUFA research was also conducted to get a better understanding on the relationship of product critical quality attributes to the in vivo performance. In this project, methoxyprogesterone acetate injectable injection was used as a model drug. Formulations with equivalent composition but different physical-chemical characteristics were prepared. In vitro drug release method were developed using different apparatus, USP4 apparatus with semi-solid adapter or USP2 apparatus with enhancer cells. The USP apparatus 4 methods show best reproducibility, and USP2 methods show better discriminated power. The in vivo PK study were also conducted using rabbit model. A level A IVIVC was successfully developed using data obtained from USP4 with semi-solid adapter. In summary, based on dose interval, indication, therapeutic duration, as well as drug release feature, injectable drug substance suspensions can be roughly categorized into 3 groups. It's strongly recommended that applicants discuss any alternative in vitro B approach through pre-ANDA meeting prior to submitting an ANDA. For groups 1 and 2 products, in vitro B options are available in some of the PSGs. It may be feasible to use in vitro B studies to demonstrate BE. If an applicant would like to propose an alternative B approach for PSG that does not include an in vitro B option, for group 1 and 2 products, supported information should be provided, including but not limited to whether the proposed formulation is Q1, Q2 the same, justifications of the selected CQA, and comparative physical-chemical categorization data. For group 3 products, although in vitro B approach may be scientifically feasible, additional information may be needed to support an in vitro B approach, such as PK data or IVIVC to support the design space of the selected critical quality attributes. GDUFA research has been helpful in developing new tools to categorize injectable suspensions to support bioequivalence or for quality control purpose. Challenge question number 1. Which of the following bioequivalence approach is currently recommended in a PSG on long-acting systemic injectable suspension that has potential safety concerns? A. Biowaiver. B. In vitro approach. C. In vivo PK study. D. B and C. The correct answer is C. Challenge question number 2. True or false? FDA recommends in vitro only B approach for immediate release suspension products like dantrolene sodium injection in the product specific guidance. A. True. B. False. The correct answer is A. I would like to acknowledge Dr. Yan Wang, Dr. David Kozak, and Dr. Rob Lineberger's valuable feedback and support when preparing the presentation. I also want to thank our internal collaborator, Dr. Xiaoming Xu, and Dr. William Smith, and external collaborator, Dr. Dan Burgess, for the great work in this area. Thank you for your attention. If you have any questions, I will be happy to answer the question in the panel discussion.
Hello, today I'd like to talk to you about a case study relating some of the challenges for in vitro testing that we observed for triamcinolone acetonide injectable suspensions. During this talk, hopefully you will gain some understanding of flocculated suspensions, how particle size relates to clinical performance, and whether your test methods are measuring what is relevant. Particle flocculation is a conscious formulation choice used to impart shelf stability, but this has been observed to potentially lead to variation in PK responses, especially for injectable suspensions. Flocculation is a process by which small primary particles in suspension will collide to form loosely held larger secondary particles called flocculates or agglomerates. This can be seen here in a polarized light micrograph showing what it looks like in practice with smaller 1 to 2 micron particles and larger flocculated secondary particles in the tens of microns. Formulating a product for flocculation is done by balancing the formulation composition. This can be done through the use of polymers, surfactants, and salts, which change the interactive forces and tune particle-particle interactions. Each of these compositional variations act as a balance, and when these interactive forces, dispersive, electrostatic, and steric, are correctly balanced, particles will exist in a secondary minimum where the particles can associate and dissociate reversibly, with a high enough repulsive barrier where the particles cannot get close enough to each other to irreversibly aggregate into the primary minimum. As this particle flocculation process impacts the observed particle size, a go-to in vitro method for measuring particle size distribution is laser diffraction. The sample is dispersed in a media and circulated through an optical cell, where the angular dependence of the scattered light is used to determine the particle size. As the breakup and collision of flocculated particles in suspension is dependent upon the shear, we can control shear experimentally a couple of ways in the laser diffraction system. The suspension first experiences shear from the stirring, which helps to keep the particles from sedimenting during measurement. The shear arises from the velocity gradient between the propeller and the wall container closure. Second, the particles will experience shear from the velocity gradient caused by flowing through the tubing in and out of the cell. Lastly, an inline ultrasonic probe can be used in addition to impart a higher degree of shearing. Here we have a figure showing the particle size distribution of a single lot of a single product under different shear conditions. As you can see, shear-induced deflocculation can cause a change in the mean particle size, with the mean particle size decreasing with increasing stir rate. Then with the addition of a low-power sonication inline, the particle size distribution broadens and shifts still to lower particle sizes. We can begin to see the presence of the signal for the primary particles arise between 1 and 3 microns. The reversibility of flocculation is illustrated here in this sonication cycling study, where once sonication is stopped, the particle D90 begins to increase. The D90 is the particle size of the 90th percentile of the distribution, which represents the larger secondary flocculated particles, with the corresponding span values seen below, which is representative of the distribution's broadness or system's polydispersity. This flocculation behavior is also directly observable via sedimentation studies. Here we see the formation and sedimentation of flocculated particles over the course of several hours. Both the sedimentation rate and the sedimentation ratio, or the volume of the sediment over the volume of total suspension, are useful correlations to product particle sizes. The short-term sedimentation ratios have a good correlation with the particle D90 size as measured under low shear conditions in the laser diffraction setup, with the long-term sedimentation ratio over, say, the course of a month correlating well to the primary particle size and total number of particles in suspension. Overall, a sedimentation study can be easily performed and can be a useful surrogate for particle size measurements. This flocculation and sedimentation behavior is actually noted in the product's package insert, where it notes that the vials should be shaken to ensure uniform suspension, and also that the injection should be performed without delay to prevent suspension from settling in the syringe. There is one other important note here, which is that only the needle length is mentioned anywhere in the administration guidelines. As we will go on to see, control of the administration procedure could be an important factor in minimizing possible clinical variability for flocculated suspensions. We have posited that a possible culprit for the observed clinical variability of these products is syringe-induced deflocculation caused by shearing during administration. As the suspension is expelled from the syringe, it travels from the larger cylindrical radius of the syringe body into a narrower one inside the syringe needle head. In order to maintain the same volumetric flow rate, the suspension must therefore travel at an increased velocity through the needle. This steep change in the velocity gradient imparts a high degree of shear to the flocculated particles as they pass through the needle, with the magnitude of the shear being proportional to the infusion rate as well as the needle gauge that is used. So what is the impact of flocculation on dissolution? From the Noyes-Whitney equation, we know that smaller particles with higher surface area dissolve quicker. If the particle size distribution for a flocculated suspension is impacted by shear, the overarching question then becomes, how does shear, both during administration and during in vitro release testing, impact the particle dissolution behavior? When designing an IVRT method, there are several things to keep in mind. Are we using sink or non-sink conditions? What apparatuses are we using? Is the method discriminant? Does the method have relevance or correlation to any in vivo performance? For an oral suspension, you might consider that the product goes from a relatively high dilution state with minimal shearing during administration, but maybe higher shear during mixing in the stomach. This is in opposition to an IM parenteral, where there is high shear during administration via syringe and a relatively low dilution and shear from perfusion in the intramuscular depot. For our experimental design, we developed a non-sink IVRT method that probes both shear during the administration as well as the environmental shear of mixing to investigate their impact on the particle dissolution. Varying the introduction method was done either via 25 gauge syringe needle or via 1 ml micropipette, as well as conducted at various stirring rates. This is an example comparison of the products which were measured using low shear conditions both in the laser diffraction with the particle size distributions shown on the right and with low shear sample introduction as well as low speed stirring rates, with the dissolution rates corresponding nicely to the observed differences in the particle size distribution. Further, if we take the same lot of the same product and simply change the method of administration from the micropipette to a 25 gauge syringe, we observe a significant change in the particle dissolution behavior, with high shear introduction exhibiting at least 4x greater dissolution rates than the low shear methods. This can also be seen in the particle size distributions on the right, where triplicate measurements of the low and high shear introduction rates show different mean particle sizes as well as particle polydispersity. To more deeply understand this dissolution behavior, we developed a novel means of simultaneously measuring the particle dissolution rate while tracking the particle size. We did this by incorporating a pion in situ fiber optic UV vis probe into our existing laser diffraction dispersion unit setup. Here we show some representative data for the low shear IVRT dissolution time course for one product. This shows how a flocculated suspension can exhibit a large particle size change over time. Beginning at the lower right hand of the dissolution curve, each point has a corresponding particle size distribution associated with it. The flocculated suspension exhibits a large shift in particle size distribution during dissolution. This is in contrast to the high shear syringe administration method, which shows a relatively small change in particle size over time and a faster dissolution rate. From this tandem particle sizing dissolution setup, we can extract some additional complementary data. Specifically, we can use the change in light obscuration, which is an indicator of the number of particles dispersed in suspension in the upper right hand, in combination with the change in particle size in the lower right hand, to make an assessment of the dissolution behavior. First, for the low shear micropipette introduction or flocculated system, it maintains a relatively constant initial number of particles with a steady decrease in particle size, which is indicative of the slow dissolution of large particles or erosion. This is in contrast to the high shear deflocculated system where the syringe was used as the introduction method, which shows a more typical behavior of particle loss over time. This has a significant implication on the dissolution rate, as the initial in vivo release of drug may be skewed due to the dissolution of either larger particles or smaller particles with different dissolution rates. So which factors do we know impact the flocculation state of a suspension? Is it A, ionic strength of the dispersant or diluent, B, the use of suspending agents or viscosity modifiers, C, agitation and temperature, or D, all of the above? Of course, all of these factors have a strong impact on the particle's flocculation state. The first two having a bigger impact on the formulation or the initial flocculation state, but agitation and temperature have a distinct impact on the distribution of primary and secondary particles. The next question is, when might you choose to use a low shear and a non-sink in vitro release test condition? Do you never use them? Do you always use them? Are you using them for comparison of secondary or flocculated particle populations? Or are you using them for comparison of primary particle populations? In this case, a low shear method is more well suited to measuring the dissolution of the secondary particle population, or the flocculated state. If you wanted to say measure the dissolution of primary particles, it would be advantageous to use a higher shear condition in your IVRT method. This may impact how well your in vitro release test method correlates with your in vivo release data. In conclusion, the flocculated states of the particles is highly dependent on shear, and therefore when deciding on particle sizing and dissolution methodologies, the shear dependence should be kept in mind. Questions like, does the route of administration for this dosage form impact the flocculation state? And if so, do your in vitro methods need to reflect this? Going back again to the example of an oral suspension versus an intramuscular injection, you can assume that by pouring your suspension into a spoon and swallowing it, there is relatively low shear. This is in stark contrast to administration via syringe. In the end, if you would like to know how the dissolution of the particles as flocculated and as deflocculated compare, it is very important to measure your particle suspensions under both conditions. This may therefore explain any variability you see in your final PK or in vivo data. I would like to give thanks and acknowledgments to the entire team that helped work on this project, including my supervisor, Xiaoming Xu, our OTR team, Zhongyun Bei and Ying Zhang, as well as our OGD colleagues, Yan, Bin, and Darby, who brought this very interesting project to us for study. For some additional resources, you can look at our entire methodology under the link to our journal article here, as well as one of the FDA newsletters concerning the IVRT for injectables, some information on intramuscular injection administration guidelines, as well as some basic reading on flocculation in pharmaceuticals, as well as DLVO theory of flocculation. Lastly, I would like to thank you all for your attention, and please don't hesitate to reach out if you have any additional questions about the study or our experimental design once you take a look. If you have any additional questions, you can contact me at williamsmith@fda.hhs.gov. And of course, I am willing to take any of your questions during the panel session. Thank you so much again for your attention, and I hope to talk to you soon.
Hi, I'm Hongna Wang. I'm a policy lead in the Office of Policy for Pharmaceutical Quality. I'm very glad to be here today to talk about the access volume map 5019.1, allowable access volume / content in injectable drug and biological products. The learning objectives for my presentation. First, understand the concept of connect container content and gross content for injectable drug products filled into vials and ampoules. After the presentation, you will be able to establish appropriate access liquid or solid filled into vials to ensure compliance with applicable regulation. You will also learn how to establish the gross content limits for finished drug products, as well as the in-process controls for bulk fills. The problem statement for injectable drug products filled into vials is that the inappropriate fill volume or content has a negative and in some cases significant impact on the patient. This is more common for oncology drugs supplied in injectable vials and ampules. It is well known that insufficient volume causes problems during administration. In such case, the healthcare provider would need to use part of additional vial to deliver the labeled dose, and then dispose the remaining drug in a second vial. Or they would have to underdose the patient with one single vial that is underfilled. On the other hand, too much of the fill volume will also cause problems because the leftover drug product in the vial can be pooled with other leftover drug to produce a second dose. And such practice may lead to drug product contamination and increased risk of blood-borne illness transmission between patients. Before we dive into the MAPP itself, let's briefly go through applicable policy on this topic. 21 CFR 201.51G states that the declaration of net quantity of contents shall express an accurate statement of the quantity of contents of the package. It further illustrates that for liquid drug in ampoules or vials intended for injection, it shall be considered to express the minimum quantity, and the variation above the stated measure shall comply with the excess volume prescribed by the national formulary or the U.S. pharmacopoeia for filling of ampoules. In the case for a solid drug in ampoules or vials, the declaration shall be considered to express the accurate net weight. The USP General Chapter 697, Container Content for Injections. The stated purpose is to ensure the drug product contains sufficient access to allow withdrawal of labeled quantity of drug. It provides test procedures on how to determine the volume of injection in containers. In addition, USP General Chapter 1151, Pharmaceutical Dosage Forms. It includes excess volume recommendations for injections based on the labeled size or the volume. It is expected that all applications should be in compliance with the CFR regulation and meet the USP requirement for container contents. Overview of the excess volume map. The map was posted on FDA's website in December 2021 and became effective on January 28, 2022. This map describes the policy and procedures for OPQ quality assessors to ensure consistent assessment for the excess liquid or solid filled into vials for injectable drug products. The excess volume should be appropriate to deliver the labeled content of drug product and to avoid excessive overfill. The scope of the map is to cover injectable drug products in vials and ampoules, including those require constitution or reconstitution. It includes single-dose and multiple-dose injectable drug products submitted in new drug applications, NDAs, biologic license applications, BLAs, abbreviated new drug applications, ANDAs, and applicable supplement to those applications. This slide lists some of the critical terms for liquid injectable drug products. Deliverable volume, expressed as ml per vial, is the volume that can be withdrawn and delivered following procedures in USP 697. Net container content, expressed as ml per vial. It also called labeled content. Is the net quantity of content that appears on the container label as a distinct item. The net container content for liquid injectable drug product will be met if deliverable volume conforms to USP 697. The gross content is the total content of the drug product expressed in ml per vial including the excess volume. This is a new term introduced in the MAPP as a drug product specification with a lower and upper limit. By implementing the gross content specification for liquid injectable drug products, the vial will contain sufficient content to deliver the labeled content and to avoid excessive overfill. Then for solid injectable drug products that require reconstitution or constitution prior to administration, net container content expressed as weight per vial, for example, milligram per vial, is the labeled quantity of drug substance or protein content. Again, this is the distinct item appears on the container label. Because the net container content for solid drugs cannot be measured directly, it will be met under two conditions as listed at the bottom of this slide. The deliverable volume after reconstitution conforms to USP 697, and the concentration of the final reconstituted solution meet the labeled concentration. In line with net container content for solid injectable drug products, gross content is the total content of the drug substance or protein content in weight per vial, including the excess amount. The lower limit of the gross content ensures that the filled amount is sufficient to deliver the net container content, and the upper limit of the gross content makes sure that the excess fill will not result in the reconstituted solution exceeding the upper limit of the assay specification. Continuing the discussion for gross content. Again, the gross content is defined as the total content filled in the vial, including the excess amount. We recommend that the gross content be assessed and released as part of the drug product specification. I want to point out here that the gross content, although it's a new term introduced in the MAPP, but it's not a new concept or something different than what you have already been doing in order to comply with CFR regulation for container content for injectable drug products. Again, it is based upon existing expectations, and the MAPP provides recommendations as how OPQ staff will ensure CFR compliance during their review. The minimum and maximum fill volume or content is a critical in-process control that says how much of bulk material should go into a vial. According to the MAPP, they should be established based on the calculated gross content limits. The minimum fill volume or content should not be less than the lower limit of the gross content, and the maximum fill should not exceed the upper limit of the gross content. This is an example for a liquid injectable drug product. In this case, the product is characterized by the label as 5 ml of a 100 mg per ml solution in a single dose vial. In this example, net container content or the labeled size is 5 ml, and the USP 1151 recommended excess volume is 0.3 ml. So the lower limit of the gross content is defined as the net container content 5 ml plus USP 1151 recommended excess volume 0.3 ml. So 5.3 milliliter is the lower limit of the gross content. The upper limit for gross content is set at 5.8 ml in this case to prevent an excessive amount of liquid in the vial. Then the acceptable minimum and maximum fill volume are determined by meeting the gross content specification. The minimum fill volume should not be less than the lower limit of the gross content, 5.3 ml, and the maximum fill volume limit should not be more than the upper limit of the gross content, 5.8 ml. So to summarize the relevant drug product in-process specifications in our case study, regarding the drug product specifications, there will be 3 separate items: deliverable volume, IC, and gross content. The deliverable volume in this case is not less than 5.0 ml per vial. IC, described in terms of mg per ml, would be the typical 90 to 110 mg per ml, or 95 to 105 milligram per ml for narrow therapeutic products. For gross content, the range would be from 5.3 to 5.8 ml. For the in-process control specification, the minimum fill volume should be not less than 5.3 ml, and the maximum fill volume should be not more than 5.8 ml. The in-process control for bulk solution IC is generally considered as part of the manufacturer control strategy to ensure that the batch is formulated with the intent to provide 100% of the label amount. Now let's look at how to apply this MAPP to solid injectable drug products. This would include both lyophilized drug products and solid-filled parenterals that require reconstitution or constitution prior to administration. In this example, the drug product defined by the label contains 400 milligrams of a lyophilized powder in a single-dose vial. As a reminder, this 400 milligrams represents the night container content for this product. The label further states that prior to administration, you reconstitute the solid with 20 ml water for injection to give a reconstituted solution of 20 mg per ml. So first thing to look at, we want to make sure that the reconstituted solution has sufficient liquid available to account for losses during administration. To assess this, we need to know the total volume of the reconstituted solution, which must be determined experimentally. In this example, the total volume was 21.4 ml, 20 ml coming from the water for injection per the label instruction, and 1.4 ml, which is called the volume of expansion. So our reconstituted drug product has an actual 1.4 ml of volume, which is in line with USP 1151 recommendations. Second thing to consider is that this actual 1.4 ml of volume should not change the label concentration for the reconstituted solution. In the equation at the bottom, the amount of API present in the vial divided by the total volume of the reconstituted solution equals to the label concentration of final reconstituted solution. This equation will be important for how we establish the drug product specification of gross content, as we will see on the next slides. Focusing now on how to determine the upper and lower limits of the gross content specification. Using the equation carried over from the previous slides, the lower limit of the gross content spec would be determined by multiplying the 20 mg per ml labeled concentration by 21.4 milliliter total reconstituted volume to give 428 milligrams of API per vial. This is the minimum amount of API that must be present at release to ensure that 100% of the net container content, 400 milligrams in this example, can be delivered to the patient. I'd like to note here that the gross content specification is part of the drug product specification assessed at release, and this lower limit is not relevant to shelf life stability. For the upper limit of the gross content spec, the math is very similar, but we use the upper limit of the IC specification for reconstituted solution, which in this case is 110% or 22 mg per ml, multiplied by the 21.4 ml total reconstituted volume to give 471 milligrams of API per vial. Shifting our attention to the manufacture process, there are two recommended approaches as how the in-process fill volume limits are established for solid injectable drug products. These approaches are not explicitly defined by the MAPP, but I'm providing them here to show different options as an applicant may choose to use. The first approach is to adjust fill volume based on the actual bulk concentration for each batch. Alternatively, a set fill volume can be used based on the acceptable variability in the bulk concentration. Using the adjustable fill volume approach in our Lyo case study, the target bulk concentration is 40 mg per ml. The actual bulk concentration tested prior to filling, for example, is 39.6 mg per ml, or 99% of the target. So if we use these in-process assay results with the defined gross content limits of 228 to 271 mg of API per vial, we will get a minimum fill volume limit of at least 10.8 ml and a maximum fill volume limit of not more than 11.9 ml. In real situation, the minimum and maximum fill volume may need to be tighter than this range limit, depending on other manufacturer control strategy established for a particular drug product and process. Then in the set fill volume approach, we use the acceptable in-process assay range to define the manufacturer fill volumes. In this example, see the acceptable bulk concentration variability is from 97 to 103 for the demonstration purpose. The gross content specification is still from 428 to 471 milligrams per vial. The only differences from the previous approach is that instead of using the actual bulk concentration in the calculation, we would use the lower bulk assay limits to get the minimum fill volume and the upper bulk assay limit to get the maximum fill volume. So in this case, 97 to 103% bulk assay range would give us an in-process fill limits from not less than 11 ml to not more than 11.4 ml. Well, as you can see, a fill volume range of 11 to 11.4 milliliter is very narrow and may be difficult to achieve depending on the manufacturer capability. Notably, as the in-process assay range widens, the narrower the fill volume limits will have to be to ensure the gross content spec can be achieved. So for a drug product with a wider acceptable bulk assay range, the applicant may consider using the adjustable fill volume approach to establish the bulk fill volumes. The bottom line is that whatever the approach you follow or you choose to use, the in-process fill volumes should be established to ensure that the gross content specification for drug product is met. Now let's test our knowledge. Challenge question 1. For an injectable ANDA application received after January 28, 2022, which of the following is applicable in terms of excess volume during assessment? A. MAPP 5019.1, but not CFR 201.51G. B. CFR 201.51, but not MAPP 5019.1. C. Neither CFR 201.51G nor MAPP 5019.1. And D. Both CFR 201.51 and MAPP 5019.1. And the answer is D. Both the CFR regulation and excess volume map. Challenge question 2. Which of the following is the drug product quality control test to ensure the maximum fill is not unreasonably large that results in leftover drug product to be used as a partial or second dose? A. Net container content. B. Gross content. C. Minimum and maximum fill volume or fill weight. D. All of above. And the answer is B. Gross content. Please note that the net container content is used to define the lower limit of the vial fill, not the upper limit. And the maximum and minimum fill is in-process control. Now let's summarize what we have discussed. Excess volume or content for injectable drug product should be appropriate to deliver the net container content of drug product or drug substance and to avoid excessive overfill that may lead to unsafe handling. Net container content is the net quantity of content as described in CFR 20151G. The declaration shall appear as a distinct item on the label, and in the case of large volume parenterals, may be embossed on the glass. Gross content is the total content of the drug product or drug substance per vial, including the excess amount. It is part of the drug product specification assessed at release to ensure compliance with CFR regulation for injectable drug products filled into vials. Well, this concludes my presentation. Thank you very much for your attention. I will be back shortly to answer your questions in the Q&A panel.
Thank you for all the great presentations. As a reminder, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We'd like to welcome to the Q&A panel Dr. Uptal Munshi, who's the Division Director of the Division of Bioequivalence 1 and the Office of Bioequivalence, OGD. He leads a team of scientists responsible for the assessment of bioequivalence section of ANDAs and other stakeholder submissions. Next, we welcome Dr. David Anderson, who currently serves as a branch chief in the Division of Microbiology 2, supporting the complete manufacturing assessment, which includes facility, chemical process, and sterility control of IND, NDA, and ANDA applications. He joined the FDA as a chemist in the Office of Process and Facilities, now the Office of Pharmaceutical Manufacturing Assessment, in 2015, providing support to a variety of activities while the office was in its infancy. And finally, on our Q&A panel, we welcome Janice Brown, who currently serves as the branch chief in the Policy Development and Evaluation Branch 1 in the Office of Policy for Pharmaceutical Quality, OPQ in CEDAR. Janice has 20 years experience in CEDAR, serving as a reviewer and quality assessment lead in the pre-approval and post-approval review divisions, evaluating product quality sections in INDs and NDAs, providing regulatory guidance to NDA applicants, and serving as an application technical lead in OPQ. Looks like we have a few questions coming in right now. The first questions are directed to Dr. Shinran Lee, and here's the first question for Dr. Lee. When we define exception excipient, is it based on the function per drug label?
Thanks for the question. The current practice is to apply the function as listed by the RD sponsor and also the ANDA applicant. And if the inactive ingredient has multiple functions and it is deferred as both non-exception or exception excipient, this would be treated as a non-exception excipient subject to Q1, Q2 assessment. Thank you.
Thank you for responding to that question. We have another question for Dr. Lee, and here is the question. What should an ANDA applicant refer for RLD products that are discontinued or for which only the reference standard exists?
Thanks for the question. So regulations require Q1, Q2 comparison to RLD formulation, not the reference standard. So applicant can submit a control correspondence to request the RLD or RS designation as appropriate. So when the RLD is discontinued, it would still need Q1, Q2 comparison to the RLD formulation, and all necessary justification are required to support the waiver request. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a few questions that came in for Dr. Ben Shen, and here's the first question for Dr. Chen. For Group 3 suspensions, is IVIVC using animal studies acceptable as a support for in vitro bioequivalence?
Thank you for the question. Yeah, for this question, you may consider using animal studies to support your product development. However, for regulatory consideration, the study needs to be. Thank you.
Thank you for responding to that question. We do have another question for Dr. Chen, and here is the question. Is the long half-life of a drug substance enough to support an in vitro BE approach for an injectable suspension for long-term use?
This is a great question. Thank you. Yeah, for the long half-life of drug substance itself, it's not sufficient to support an in vitro BE approach. In vitro BE approach is a totality of evidence approach that requires strong scientific justification, covering considerations on critical formulation characteristics, drug release mechanisms in vitro and in vivo. If you would like to propose an alternative in vitro only approach to the agency, for example, through pre-ANDA meetings, you should provide sufficient support information. For example, you should provide whether your proposed formulation is Q1, Q2 to the RLD, and what are the formulation characteristics you propose and the justification, as well as data of comparative physical-chemical categorizations. A little bit special for the Group 3 product is that in addition to those information, we recommend you also provide data to support the design space and relationship that can help justify the proposed physical-chemical categorization in IVR2 to support the equivalence. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a couple of questions that came in for Dr. William Smith. And here is the first question for Dr. Smith. The differences in dissolution during the first 5 minutes of dissolution will not matter so long as the Q value is met by both of these formulations. So why should it matter for the in vitro method?
I think that's a good question. The problem is that if you say a Q value of 75% of drug dissolved in 30 minutes, and within the first 5 minutes, one product releases 50% and the other one releases 10%, but they both reach the same Q value at that 30-minute mark. For things that have high solubility and high absorption, as far as like ADME, what we've noticed is that in the PK data for these kinds of products, that you can actually see some bimodality where you get an initial spike and then you get the expected release. So I guess that would be kind of the
the issue with saying, oh, they both reach a certain Q value at a specific time, but how they get there might actually impact the in vivo performance. Thank you.
Thank you for responding to that question. We've got another question for Dr. Smith, and here is the question. Shouldn't PSD measurements always be targeted on measuring primary particles? In your present case, injection mode, low or high shear, influence the measurement result. Shouldn't a valid LD-PSD method deflocculate suspension samples via mixing and ultrasonification without the need of a high shear needle injection?
I think that's a good point. Whether or not we decide a method is valid is really up to the 3 R's of repeatability, reproducibility, and robustness. So you can really measure it under high or low shear conditions. In this example, I think it's important to remember that from previous studies, what we saw was that the primary particle size for a flocculated suspension doesn't correlate well with the in vivo performance for a flocculated suspension, but the secondary particle size does seem to correlate better. Whether or not you should measure one or both is up to the company as far as determining for BE purposes how they compare to the RLD product. But I think that's the takeaway, is the correlation.
Thank you for responding to that question. Moving on to our next panelist, we do have a couple of questions for Dr. Hannah Wang, and here is the first question. For MAPP 5019.1, allowable excess volume, content in injectable drug and biologic products, what applications or submissions will be affected by this MAPP?
Yeah, the assessment for applications submitted after the MAP effective date would follow the recommendation outlined in the MAP. While the MAP does not apply to pending applications that were submitted before the effective date of this MAP, all already approved application. However, please note that all applications are expected to be in compliance with the CFR regulation for container content or well field size. If approved application and pending applications submitted before the January 28, 2022 are not in compliance with CFR regulation, the application holder should take the appropriate step outlined in the applicable regulation and FDA guidance to bring their application into compliance. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we did get a question in for Dr. Uptal Munshi, and here's the question. When there's a discrepancy in the Q formulation or information for an RLD through labeling and packaging text, which is detected during CC between an end applicant and the FDA, is there a formal follow-up action by FDA to inform the RLD manufacturer to correct such discrepancy? If the RLD is withdrawn and not available, what is the mechanism for CC for Q1, Q2 based on the RS product's quantitative composition?
Yes, thank you for that question. So I guess let me start with the second question, or the first question related to discrepancy. If you believe that based on the results of your reverse engineering, you think that there's a discrepancy, then we would encourage you to submit a control and share that information with the agency for consideration. when assessing your proposed generic formulation for Q1, Q2 sameness to the RLD. Such information will help ensure the accuracy of product labeling information and support generic drug development. We continually strive to improve our review processes and monitoring of FDA-approved products, and your input will help ensure that drugs are safe and effective for use. As it relates to the second question of the RLD being withdrawn and not available, we note that as a regulatory matter, the Q1, Q2 comparison should be performed against the RLD product. We understand that there may be limited information available publicly with regards to the RLD formulation. However, we encourage you to submit a control correspondence to seek a Q1, Q2 assessment of your formulation against the RLD before submitting your ANDA. The control correspondence pathway is intended to help applicants with developing their specific drug product by providing feedback on various regulatory requirements, including inactive ingredient sameness to the RLD. We also recognize that due to formulations being trade secret information, FDA's responses to your Q1, Q2 assessment questions are not always enough to resolve your formulation questions to achieve a Q1, Q2 same formulation. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a question for Dr. David Anderson, and here is the question for Dr. Anderson. What test procedures do you recommend to be included in the liquid drug product specification for gross content in order to release a batch?
Hello. For a liquid drug product, an in-process control for fill weight measurement and the corresponding sampling plan may be used to satisfy the gross content specification. The testing plan and the acceptance criteria for gross content would be included in the drug product specification table with a footnote to indicate that it is tested in process. An applicant could also, under ICH Q12, list in the in-process testing and associated acceptance criteria as an established condition used to meet the gross content recommendations at release. In order to release a batch, the gross content would then need to be assessed by reviewing the executed batch records to ensure the batch data for in-process weight testing is acceptable. Note that these are just some examples of recommended test procedures, and alternative strategies may also be adequate with justification.
Thank you for responding to that question. We do have another question for Dr. Anderson. Here's the question. What test procedures do you recommend? Oh, I think it's been repeated twice. Sorry about that. Let's see, we have another question for Dr. Anderson. All right, going on to our next panelist. We have some questions that just came in for Janice Brown, and here is the first question. Is net container content a separate test required in the drug product specification? All right, Janice, we're having a little bit of difficulty hearing you. What we're going to do is go on to our next panelist. We're going to let the tech team reach out to you to adjust your volume and then check back in with you. Moving back up to the beginning of our panels, we're bringing back on Dr. Shenwen Lee. And here's a question for Dr. Lee. Is a waiver under Regulation 314.99b the same as the waiver under 320.22?
Thanks for the question. So waiver under 314.99b is not the same as waiver under 21 CFR Part 320, for example, 320.22. 314.99b is a waiver provision And the 31499B waiver request is to seek waiver of inactive ingredient requirements. So when applicants requesting a waiver of in vivo B study requirement, a separate wait request is required, as the requirements for bioequivalence are governed by the regulations set forth in 21 CFR Part 320. Thank you.
Thank you for responding to that question. We still are checking in with Janice Brown. She's not having any audio right now, so we're going to try to move on to another question for Dr. Lee. And here's the question for Dr. Lee. Regarding example case 1, could you please clarify if exception excipients, buffer, and antioxidant can be replaced with a non-exception excipient like a tonicity adjuster?
Thanks for the question. So the case 1 is for the proposed formulation, which is not Q1, Q2 the same to the ROD in exception excipient. Under 21 CFR 314-94A93, a variation between test and ROD product is allowed for parenteral formulation with respect to buffer, preservative, and antioxidant. So the tonicity adjuster is not an exception excipient. If the test formulation is different from the ROD with non-exception excipient, then other supportive documentation must be submitted by citing appropriate waiver provisions. And then the acceptability of such waiver requests will be determined during the scientific review of the ANDA. Thank you.
Thank you for responding to that question. We just have a few more minutes left. We're going to move down to Dr. Hannah Wang, and we have a question, the following question. Is the net container content a separate test required in the drug product specification?
Thank you for that question. No, net container content test is not a separate test in the drug product specification. But the applicant should provide data to demonstrate that their drug product can meet the requirement for net container content. As stated in the MAPP, for liquid injectable drug product, the net container content on the label will be met if the minimum fill volume conforms to USP General Chapter 697. And for some product, the net container content on the label will be met under 2 conditions. First, total volume after reconstitution conforms to USP General Chapter 697 following the reconstituted procedure in the labeling, and also the labeled concentration following the reconstitution procedure as described in the labeling is met when the vial is filled with the minimum fill content or volume of the bulk drug product. Thank you.
Thank you for responding to that question, and we're all out of time for questions during this round. We want to thank all of our panelists for answering numerous questions that came in and for all the great presentations. Well, we'll now head into our break.